these topics that we have had, the symbiosis is a really important element for innovation, of evolution of life. And probably you can recognize the only thing that we all already recognize is that symbiosis was so important that otherwise we will not be here, right? All the eukaryotes will not exist, and the part the bacteria that first start to create this atmosphere and then be part of mitochondria and the plastics in the plants that we know symbiosis has delivered a lot of innovations. So of course, right now, we are all aware, and there's a new concept that I really like, because I really like symbiosis. And so I think this is a very important concept that we have to keep in mind. Probably some of you already know of this, and it's the holobiome concept. And probably this holobiome concept actually says, or relies on the following evidence that I will show you today. Well, most animals and plants contain abundant and diverse microbiota. And this is something that we know because of this tree of life, that microbes were first here, and then eukaryotes emerge. So, of course, some of the work that I have done before have also shown that even fungi can also be associated with all the microbiota. In the talk of today, I will talk about the plants, the can plants. So I will show you that, of course, plants do contain diverse and abundant microbiota, but this is probably not so difficult to imagine. What is more important for biology is actually that this symbiotic microbiota is affecting the fitness of the host, the survival, and sometimes even the distribution of the plants or the animals, even the behavior. And the second, third line of evidence is actually that microorganisms, symbiotic microorganisms, can be transferred from the parents to the offsprings. So of course, we are here in the microbiology session. We don't think microbes are really central to life and to biology. But of course, this evidence actually suggests that we cannot consider plants and animals as individual entities, no? They are not separated from the microbes. So this holobiome concept actually takes into account that when we see an animal or a plant, what we are actually seeing is an eukaryotic host and a lot of microbial symbionts that are associated to it. And by this association, they actually create an emergent individual. And probably you know the definition of emergent properties. Emergent is also a characteristic that is only it only takes place when the things are coming together. So if we take apart all the parts of the system, we will not see the phenotype that is actually created by the combination of microbial symbionts and the host. And of course, now that we are in the genomic area, we can say there's still an whole genome. And this is this can refer to the eukaryotic host genome. Of course, the organelles, we know the mitochondria and the plastids in the plant, they have their own genetic information. And of course, we have a very wide pool of microbial symbiotic genomes. And all of this is very important for evolution and something that we would like to test also in plants. Of course, the task is not so easy to go through these lines of evidence that I have shown. So, something that I'm really convinced as well is that by asking fundamental questions, we can also resolve problems, right? So I don't think fundamental research is struggling against using applications. So one of the things when I get back to Mexico after being in Europe for some time, I realized that by looking at a map, that 1/3 of the world surface area is actually arid and semi-arid ecosystems. And these ecosystems, actually what is limiting is the availability of water, right? Water is not abundant, and the temperatures make that less abundant. So, especially for Mexico, this represents almost half of the territory. So, we are here in Aguascalientes, and I was spending some time in Monterrey. Northern Mexico, you know we are in an arid and semi-arid environment surrounded. But especially for the agriculture, this is very limiting. And many forecasts for the 2100 says that because of global warming and all our human activities, actually the drylands will increase up to 50%. So of course, this imposes a lot of different challenges for not only for us, that we will have to deal with probably less area that we can really use for cultivation, but of course a lot of animals and all life. So, in these ecosystems, you know already, plants like agaves and cacti already adapted to these ecosystems. No? We know them, we see them all the time. But something that we didn't know, and we know that they have some biochemical innovations. One of these innovations is the CAM, the crassulacean acid metabolism. That is a way of photosynthesis that allows these plants not to lose so much water during the day, that is very hot, and the water that they have, they can keep it, and they fix carbon dioxide in the night. And by doing this, of course, they have succulent bodies that allow them to have water, and very superficial roots for any humidity that is in the soil, that it can be taken by them. But of course, until now, actually, we didn't know so much which kind of relationships do these plants establish with microbes. And of course, I think, based on the holobiome theory and many symbiotic examples that we have, that actually microbes do have a role in the adaptation and to these kind of environments. So, of course, you know by now, cacti is a very important plant family that originated only in the American continent, once the continents were separated. So we have cacti from the south part of Canada until the Patagonia. And of course we know as Mexicans, that well, most Mexicans, of course, we have a lot of guests from different countries, but you know, we know cacti have been very important, even for the foundation of Tenochtitlan, and probably some of people know also peyote. How many of you know peyote? And we have really an extraordinary diversity of this plant family. More than 2,000 cacti species, and the highest of diversity is in Mexico. So of course, it's an important resource for us. And what we can tell about agave. Agave is actually one of the most important plants that was used even from the Mesoamerican times. And tequila is really one of the most known things about Mexico in the world. Now we have around 200 different species of agave, and of course most of them, well, Mexico is considered to be the center and origin of these species. So we have also the highest diversity of it. So, and of course we have bichotes and all these kind of things. And they are very diverse and very resistant plants. So, what we asked is actually a very ecological question, right? If you see, most of these, well, these two species actually inhabit, well, all these arid and semi-arid ecosystems, they don't belong to the same. Well, one is a monocot, agave is a monocot, cacti is a dicot. They both produce flowers, they are angiosperms, but we didn't know much about what kind of microbial communities are associated with these plants. So we actually, if you see these two cacti and agave living together, we wonder which kind of association do they have with the microorganisms, and of course if they will be different communities because they have different genetic background. And of course what we really would like to know is how they affect the functionality and if they contribute to the adaptation of these plants to the environment. So it's very important for ecology or microbial ecology to have a very good strategy. No? So I will tell you about our strategy to deal with these questions. And of course, first, it's always the capital, the human capital. So now that I'm talking to a lot of different students, of course, you are the most important part of any research project. No? It doesn't matter how much money or equipment, really the person that are behind are the most important, the ones that are curious and can look for really different alternatives. So I was very happy to have two great PhD students that really joined my lab when it was really empty at the beginning, and also a very good collaborator in California, David, who together started this adventure. So we pick how to address this question. Okay. So we selected 5 different species of camp plants. As I told you, well, we were interested in agave and cacti, because they are important but very diverse. And we actually picked agave desertic, which is the plant that is in the northern part of, well, in the distribution area, that is in California and Arizona. Is one of the agaves more adapted to the driest environments. Agave tequilana is the only species that is cultivated and is the only one that is used for the production of tequila. So you don't find agave tequilana almost in wild populations. They are all grown in the tequila, for the tequila industry, in different fields. And we have agave salmiana that is also used for mezcal, but it's also, there are native populations of agave salmiana. And we also picked Myrtillocactus geometrisans, garambullo. You can eat also these fruits, and Opuntia robusta, which is actually a very important. Well, Opuntia, you know, they are very important, very big family, subfamily in the Cactaidea. So we also were interested, well, these plants, even if they occur in many different species, they are not distributed all over. As I already said, we picked 3 different places in California, in the Gold Canyon. We also have 2 different sites in central Mexico, in 2 different tequila fields, in Amatitlán, Jalisco, Herradura, Pénjamo, Tequila de Pénjamo. And of course, we have the opportunity to have 1 agave species, agave salmiana, and 2 cacti in Guanajuato, in the northern part of the country, the state. And of course, because these plants really suffer like 8 months of dry period, when there's almost no rain. So we were interested in looking if the seasonality that they suffer, like these 8 months of dry, and then 4 months of different raining periods, this will affect also the assembly of the microbial communities. And probably a lot of the people that have focused on interactions, plant microbe interactions, they have focused mostly only in the soil and the rhizosphere. But we have we applied a very systematic way, a full approach, because we also investigated the communities that are above the ground, like the phylosphere, the leaf endosphere, also the root endosphere, the rhizosphere, and the soil close to the roots, and bull soil, that is the soil 1 meter away from the plant. So by combining molecular methods and culture-dependent methods, we actually are trying to really look which microbes are really associated with these plants. And of course, we know plants are a unity, no? So probably some applications here will have impacts down and the other way around, right? So that's why we try to cover all the plant, right? So by the molecular part, we started using a fingerprinting method because it was a new lab and we didn't have so much money. So we used also the 16S and then DGTE. But later we were able to establish a collaboration with the Department of Energy in the US, and they made for us a lot of the Illumina sequencing from the 16S and the B4 region, and also the ITS. So we're also looking not only for bacteria and archaea, but also for fungi. So because fungi is also very important for most of the plants. And we know actually that mycorrhizas are the most important or the most known symbiosis of plants and microbes. So by doing this, of course, we have different factors, and of course we need statistics then to evaluate the results. So this is how it looks one of these sites, for example. So the first striking thing that we really found, by telling all this, actually we had more than 400 samples, and we sampled really 66 individual plants across this sampling that I showed you, experimental design. And by doing this, at the end, we have been able to identify around 4,000 prokaryotic and fungal OTUs associated to plants. And here I'm showing you a diagram. On that diagram, you can see the different species, and something that we can really look through this diagram is actually that the core microbiome, core are all the taxa, the operational taxonomic units, that are shared in a different, well, in the environment. In this case, that are shared by these 5 species. And we see that even the numbers are pretty similar between the prokaryotic and the fungal taxa, fungal OTUs. Actually, what is shared is much higher. 3,000 are shared by all 5 species. But in the case of fungi, we see really a lower amount. And that already is telling us that fungi is more specific than bacteria. Bacteria have a wide distribution. But of course, the first question, we wanted to know which rules, assembly rules, do these communities follow. And by doing non-multidimensional scaling, we can, this is the 16x results, and I will guide you to this slide. Actually, what we are comparing is the different taxa that are associated to a sample. As I showed you, if they are very close together, they mean that they have similar symbionts, well, similar taxa associated, and if they are separated but far away, they will be different. So one pattern that we can see here is actually that the soils, you see here, all the soils, even the soils and the soil close to the roots, they are clustering together. So that means that the soils in California, the soils that are in Guanajuato and in Jalisco, they really share most of the taxa, and they are clustering here together. As we move in this x-axis, we see that the communities are starting to separate. So we see here in red the rhizosphere, more close to the soils, and then the phyllosphere. And later we see the endosphere. So all the bacteria and archaea that are living inside the plants. And we see here the roots and the leaf endosphere. So this analysis, together with the statistics that we applied at this Termanova, we could determine that the plant compartment is really guiding the assembly of the communities. And that species and the interaction of the species with this plant compartment actually contributed to 86% of the total variance in the samples that we did. So this is very good, and considering that we did this in natural populations, right? And that we just sampled. So this is like, I didn't mention up to now that we didn't find so much archaea associated to these plants. The archaea were more abundant in the soil, but not so abundant. And actually there are 4 phyla that are associated with can plants. This is actinobacteria, proteobacteria, firmicutes, and bacteroidetes. So the next question, how the fungal communities look like? And this is using the same DNA. We amplified the ITS2, and this one looks really different compared to the ones that I just showed you about bacteria and archaea. We can see here, if you follow me, we here very distinct in the X days. We see here all the, in the right side, we see all the samples that come from California, from Agave deserti. Another one that we have on the left side, and all the samples that we made in Mexico, even from Guanajuato and Jalisco. And the other difference, very important difference, is that we see that the clustering of all the external communities is not so separated as it is by bacteria. And then what we clearly see separated is the endosphere, the ones that are living inside the plant plants. So these are different compartments, and it was also supported by the Termanova, who actually told us that fungi have a biogeographic pattern. So the soils in California have a different fungal communities, and then the plants look different because of the difference in the soil, and so the ones that associate to the plants. So, of course, once we know the different assembly rules that agave and cacti follow, we actually asked as well if cultivated agave tequilana had a reduced microbial diversity. And you can see by here using the Shannon index, actually we are showing in this case just the data for agave. We see that the soils are not different, even if they have some agricultural management. But what we did see is actually that the prokaryotes are really, in the rhizosphere and phyllosphere, they are really reduced. No? The diversity is significantly reduced in comparison to the native agaves. But something that is also striking, and that was not previously seen in other studies of microbiomes in plants, is that the phyllosphere is actually as diverse as the rhizosphere. So something that most people think is actually that the rhizosphere is the most diverse interface of interaction between the soil communities and the plant. But we see here that the cow plants actually have very high values, exactly the same as the phyllosphere, the aerial part. And this is something that is really striking, because if you imagine you are in the Sonoran Desert or in any desert, of course the radiation is much higher in this, and the fluctuation of temperature as well. So these communities have to survive these conditions, but they are still very diverse. In the case of fungi, we didn't see a clear impact on microbial diversity in between the native agave and the native species and the cultivated ones. You see, they are more or less fluctuating, and there is not a clear trend that cultivation may have an impact on these communities. Of course, the same, when we evaluate the cacti communities, it was exactly the same. The phyllosphere did have a higher diversity, and lower was in the lithosphere. And in fungi, we see that there is not so much difference between microbial diversity in the soil and in the rhizosphere. There are not so much changes about this, and just the endosphere and the phyllosphere have a reduced microbial diversity. So, of course, one of the key questions is for us was to, now that we know that the plant compartment is a very important, is the most determinant for looking who is there, no? So we can, in the case where we could compare the three different species that were in the same, sharing the same habitat, sympatric. So we have Agave salviana and these two other cacti, and you can see, well, here we are showing the rhizosphere, the root endosphere, the leaf endosphere, and the phyllosphere. And you can notice by the different colors, the different bacterial taxa that are associated with these groups. And what we can see for this one, even sometimes the relative abundance changes significantly, some other times not, but we see the same groups colonizing the rhizosphere. So this means for us that actually the sympatric plant plants do share the same symbiont. And something very interesting, as I probably remember, the phyllosphere that I showed you that was really also very diverse. The communities that are in the skin or in the leaves of camp plants, they have a cyanobacteria. Cyanobacteria, probably this cyanobacteria is promoting the establishment of other kind of heterotrophs and even fungi. So we think it's very important, and probably something that we will continue investigating. In the other case, in fungi, I didn't mention, but most of the fungi that is associated with camp plants are Ascomycota, 90%, and we have few Basidiomycota. We didn't find a lot of evidence on mycorrhizal fungi, and this is something strange, but we didn't find really they are 1% in the total sequences that we have analyzed. But something interesting is the only difference here is actually that fungi seems to have most specific patterns in the endosphere. So probably you can see this graph, this part of the graph, where we see that the three different species that are sharing the same space do have differences in the fungal community. So it seems that host specificity is acting one of the endosphere, endophytic fungi that goes well associated to these plants. Up to now, what I have shown you, that you can, the prokaryotic communities they follow, they are influenced mainly and mostly you can explain by the plant compartment, while fungal communities are affected by the geography of the plant host. And you can still think that microbial diversity associated with plant plants is very high in the soils. It is reduced in the episphere, the rhizosphere and the phytosphere, and then it's really much reduced in the endosphere. It's the lowest level of diversity. And of course, this is something that we expect because plants have also some systems to detect pathogens and to act. So very few plants really undergo or this compartment. Something interesting to follow is the similar diversity in the rhizosphere and in the phyllosphere. And as I showed you, even we don't know if this has a negative impact on the fitness of agave tequilana. I showed you that diversity is lower for this cultivated species, and we speculate that this could be related to the way people handle the agronomical practices that agave tequilana has in the fields. And of course, the native agaves and cacti do not have this, well, they have similar levels of diversity and higher than agave tequilana. And sympatric plants share, this is something very interesting for us, of course, because similar symbiotes means that we will be able to find probably specific functions that are shared in this environment across different plants. And the only thing that have, well, specificity is the endophilic fungi, and we have been able now, of course, because we have a collection of these, some of these strains, to biochemically identify that most of the strains that we have in the collection, they do have different traits, and the canonical traits for plant growth promotion, like nitrogenase, for example, nitrogen fixation, C04, the production of different hormones, and of course, tolerance to drought. So something that we wanted to start doing is actually to use this collection of microbes that we know they are associated with these plants for as potential biofertilizers. And we actually wanted to use agave plants and cacti plants that we have propagated in vitro to also use this kind of experiments. But as I showed you here, one interesting result that we have made these inoculations in a field because we have some collaborators of the tequila industry. And we have seen, well, by using more than 1,000 plants, we have collected only after 1 year that we inoculated these plants. They were already 2 years old. We inoculated different combinations of strains, and we then take the leaves and we make a metabolic profile. And very interesting, metabolic and metabolomic. Something that you can see from this heat map is actually that all these leaves were not inoculated with any bacteria. They were just mock or with urea inoculated. But we see some patterns where we have changes and a grouping of spatial consortia. So that means that some of these bacteria, when we added their cells of one fungi, they can modify the metabolic profile. And of course, up to now, some of these treatments have really rendered very wealthy and healthy plants that seems to be more vigorous, and that the producer really like us to continue doing this inoculation for him, right? And of course, one of the key questions that we will have later is actually if the strains and these plant microbe interactions that we discover will be functional for other plants that are not become metabolism, in this case tomato. And of course, working with microbes is very different than working also with plants. Plants have also a lot of plasticity. And something that I can show you just here is that these interactions really modify, for example, in this case, root architecture. These evaluations of, for example, you can see mock, how the tomato and root look like when you have a commercial biofertilizer that is already available in the market, and with one of our strains derived from cactus, we really see a more robust radical system. But of course, health is not only to have roots, so we have to compare other traits to really compare that it can be possible. But something that we really would like is, well, that we are moving in the next year, is actually to link how this diversity reflects in function. And for this, we have still support of the GTI, and we have sequenced, or about to finish the sequencing of 40 different bacterial genomes of the ones that we have already characterized. And as I told you before, also with metagenomics, it's possible not only to see who is there, but which kind of gene do they have. So we are comparing right now the differences between the microbial communities in the rhizosphere and in the phyllosphere. And we, of course, because of the presence of different groups, we expect as well different functions that the microbes are having or establishing with the plants in these two compartments. One of the key questions that we are addressing right now, something that we realized in these last years, is actually that it's difficult to work with agave cacti. One of the reasons is that we don't have any molecular tools to modify them, and of course they need sometimes one year to really show some growth. So if we cannot do it so much, we need to have results for publication and to be evaluated and to get some funding. So that we are trying to use Arabidopsis as a plant model system to understand fundamental mechanisms of the communication between microbes and plants. So the work that it has started as a PhD student in the lab has to be with this communication. Is there any signal from the microbes that is perceived by a plant that promotes growth? So you can see in these petri dishes that are divided, you see here Arabidopsis. It's very easy to cultivate, even in a petri dish. So you find it here. And here is nothing in the other side. So it's just medium, normal medium. But what happens when we cultivate one of these microbes that we have characterized from agave cacti? If you place a lot of this saturated, you see that the Arabidopsis is not feeling so well. No? It's only perceiving the volatiles from this bacteria. But if we control the doses of these bacteria, at the end we see a positive effect on Arabidopsis. And you can see it very clear here. No? You see a lot of foliar growth and more roots. So of course there are some metabolites, in this case volatiles, is what we are analyzing, that are triggering different responses in the plant. So, of course we are, well, David is already very good at managing the GC chromatograph, and he has he's identifying by these different conditions when it's saturated, when you have everything we know in chemistry is actually dose dependent, right? So we do now the question, if you would like to know more about this project, I invite you to visit his poster. Is on Thursday, no? In the session of microbiology. One of the other projects that is ongoing, of course, something that we really need to know is if there are microbes associated to the seeds. If you think that some microorganisms are in the seeds, of course they will be vertically transmitted, and this is the work that Victor is doing. We have many of them, some of them also related from the seeds of cacti.